Welcome to our greenhouse management crop evaluation from fall of 2024. This was conducted by me, Patty Sviracek, and my lab partner, Abby Cushing.
This semester, we grew a variety of crops, including Helleborus, Coreopsis, Hosta, Sedum, Echinacea, Monstera, Gara, Snapdragons, Gerbera daisies, Dianthus, 2 cultivars of Huchera, and 8 cultivars of poinsettias. We potted the poinsettias up on August 14th in 6-inch tall pots and the other plants on September 9th in 5-inch tall pots. The fertilizer we used was Jack's Nutrients 15-5-20. The concentrations were different for the poinsettias, which got a ppm of 300, while the others got a ppm of 150. This is due to poinsettias having a higher EC requirement by double. We applied the fertilizer through drip emitters, as you can see circled in blue in the picture. On August 28th, we pinched the poinsettias to promote lateral growth and identified spider mites. To treat the spider mites, we applied a granular insecticide called Marathon. We applied 1/3 tablespoons to each plant manually. On September 8th, we identified a magnesium deficiency through symptoms of interveinal chlorosis and leaf curling. We treated this by applying Epsom salt containing magnesium. To prevent future deficiencies, we continued applying the Epsom salt for a further 4 times. An advantage of using Epsom salt is that it prevents fruit shock and gives a fuller, bushier appearance.
On October 7th, we applied the plant growth regulator Bonzi. For poinsettias, it is necessary to apply a plant growth regulator, otherwise it will grow too tall. We applied it as a drench at a concentration of 15 parts per million, applying 4 fluid ounces to each plant. We found our EC levels to be far too low for what we had originally expected them to be. A few potential causes are either we made inaccurate measurements when making the fertilizer solution and setting the dosatron, or we messed up one of the steps in the process of measuring EC. The plants did not show any signs of nutrient deficiency, and most of the EC measurements that were taken were extremely close to levels of deionized water, which is what we poured into the plants during the test. This told us that more than likely the injection dosage was correct, but what we were measuring and how we were measuring was incorrect, and what we were measuring was not at the actual EC the plant was receiving, but more than likely the EC of the water we had just poured in, simply a matter of human error. We also had a few pathogenic problems pop up in the last few weeks of growth before the sale. We had a case of botrytis on the daisies that was found 2 weeks before the sale. The signs consisted of gray sporulation that looked like mold on the flowers in between the petals. To treat this, we simply cut back the flowers that were infected, along with any flowers near the infected plants. And our plant growth professional applied a drench to stop any future signs or symptoms of botrytis. The second issue we had come up was a case of bacterial root rot on the poinsettias, which was found 1 week before the sale. The symptoms consisted of leaf curl with sudden collapse of the branch. To treat it, we removed any infected tissue and reduced the days that the poinsettias were being watered, as we concluded that this disease came from overwatering. This first figure depicts the average temperature in degrees Fahrenheit over time, and it shows a decrease in average temperature. This is predictable, as during this time of year, the outdoor temperature will fall, which has an effect on the temperature inside of the greenhouse. For our operation, it was important that the temperature did not fall below 60 degrees Fahrenheit. And while it came close to that level between October 11th and October 23rd, it never fell below 65 degrees Fahrenheit, so there was no concern of damage from cold. This next figure depicts the average relative humidity and percentage over time, and shows an initial overall increase in humidity with a dip between October 11th and October 23rd. This was to be expected, as when the air temperature decreases, the water vapor holding capacity also decreases. As the plants grew larger, they transpired more, and since the average had increased over time, We know that in the future, we could possibly reduce how often we watered as when the plants matured more and the root systems grew, they needed less of the frequent watering. This last figure depicts the average vapor pressure deficit measured in kPa over time, and it shows an overall increased average vapor pressure deficit. This tells us how dry the air is, and it generally fluctuated over time. Too low of a vapor pressure deficit leaves plants susceptible to disease and more attractive to pests as the plants are stressed out. And each of the extreme dips correlated in the graph were connected to the times that a pest or disease was noted and required treatment. Preventing dips in the future by increasing greenhouse temperature via heaters can help prevent disease and make the plants generally stronger.